Hello everyone, I am Layli. Today, I would like to introduce a review article from our team published in iMetaOmics, entitled Bile Acid Metabolism and Signaling in Human and Animal Health, a One Health Perspective. The One Health concept emphasizes the interdependence of human, animal, and ecosystem health. In this context, bile acids are host microbiota co-metabolic products that function beyond lipid emulsification. This review summarizes bile acid biosynthesis, enterohepatic circulation, microbiota-mediated modification, and receptor signaling through farnesoid X and Takeda G protein-coupled receptor 5. We discuss how bile acids regulate metabolic, barrier, and immune homeostasis, and how their profiles are shaped by population diversity, circadian rhythm, diet, behavior, environmental exposure, and surgery. From a One Health perspective, bile acid-targeted strategies have translational value for human disease and animal production. By improving animal health and reducing excessive drug use, residues, and antimicrobial resistance risks, bile acid-based precision nutrition may support food safety and ecosystem health. Cholesterol is converted into Ca and Cis-Ca through the classical or alternative pathways in hepatocytes, followed by conjugation and secretion into the intestine, where bile acids facilitate lipid absorption and undergo enterohepatic circulation. A small fraction of bile acids reaches the colon and is transformed by microbial enzymes, including BSH, HSDH, T-alpha dehydroxylases, into DCA, LCA, and related derivatives. These metabolites regulate the balance between Th17 and Treg cells. 3-oxo-LCA and iso-LCA suppress Th17 differentiation, whereas iso-CA and iso-allo-LCA promote Treg generation. Their immunological effects depend on bile acid species, local concentration, target cell type, and disease context. After meals, bile acids are reabsorbed in the intestine through ASBT, exported via OSD-alpha-beta and MRP3, returned to the liver through portal circulation, and taken up by NTCP. Hepatic and intestinal FXR activation induces SHB and FGF19-15 FGFR4 beta-klotho signaling, thereby suppressing CYP7A1 CYP1 and maintaining bile acid homeostasis. Bile acid-activated FXR and TGR5 coordinate glucose and lipid metabolism by inhibiting gluconeogenesis and lipogenesis, promoting glycogen synthesis, GLP-1 secretion, Fa-beta oxidation, and TG clearance. These mechanisms are linked to adaptation to negative energy balance in dairy cows, reduced fat deposition in pigs and poultry, and alleviated hepatic lipid accumulation. In the intestine, FXR strengthens tight junction and mucin barriers, whereas FXR TGR5 signaling suppresses NF-kappa B and LRP3-mediated inflammation and promotes M2 macrophage polarization, supporting barrier integrity, immune homeostasis, and production in pigs, poultry, and aquatic animals. Bile acid metabolism is influenced by population diversity, circadian rhythm, diet, behavior, environmental exposure, and surgical intervention. Different physiological and pathological states, including diabetes, metabolic dysfunction-associated steatotic liver disease, MASLD, intrahepatic cholestasis of pregnancy, early life, and healthy aging, are associated with distinct bile acid profiles. Circadian rhythm and dietary patterns further modulate bile acid composition, microbial transformation, and host metabolic responses. Smoking, exercise, mental status, and temperature exposure reshape bile acid-related signaling in a context-dependent manner. Surgical procedures, including sleeve gastrectomy, Roux-en-Y gastric bypass, and cholecystectomy, remodel postoperative bile acid pools and downstream physiological outcomes. Blue arrows represent an increase, and red arrows represent a decrease. Future studies on bile acids should advance from mechanistic characterization toward precision translation by integrating bile acid derivatives, receptor-targeted molecules, stratified intervention, high-sensitivity bile acid detection, and clinical translation. Multiomics, organoids, isotope tracing, humanized animal models, and long-term clinical cohorts will clarify bile acid microbiota host causality, identify responders and non-responders, and support personalized nutrition, therapeutic development, and sustainable animal production. Summary: bile acids act as host microbiota co-metabolic hubs linking human, animal, and ecosystem health. Bile acids regulate glucose and lipid metabolism, intestinal barrier, and immune homeostasis through receptor signaling and microbiota-derived metabolites. Bile acids are dynamically influenced by population diversity, circadian rhythm, diet, environmental exposure, and surgery. Bile acids provide therapeutic and nutritional strategies for human disease intervention and animal health, further supporting food safety, sustainable development, and One Health benefits. Thank you.